weeks ago when we were talking about our cell structures, right? We talked about the plasma membrane. We talked about how the plasma membrane is in both our prokaryotes and our eukaryotes. We talked about how it was this barrier between the outside of the cell and the inside of the cell. And we talked that it was made up of what? What makes the plasma membrane? Phospholipids, so the phospholipid bilayer. And what else does it have? Proteins, right? So we have phospholipid bilayer, and it's going to be associated with a bunch of different kinds of proteins. Remember all of those fun proteins? We were making them in the rough ER, and they were getting modified in the Golgi, and they were shipping out to the membrane proteins. They're membrane proteins. Here they are. All kinds of, well, I'm going to point to this. So listen, point. All kinds of fun membrane proteins. We have peripheral proteins that are just going to be associated with either one side of the membrane or the other. Proteins can be associated with the inside leaflet, we call it, of the membrane, or they can be associated with the outside. But they can be associated with the outside of your membrane. You can also have integral proteins. Integral proteins are going to go all the way through your membrane, through both leaflets of the phospholipid bilayer. You can have other kinds of substances as well. So you can have things like glycoproteins. These are going to be proteins that are associated with carbohydrates, and we talked about them a little bit in terms of things that your cell might be making as well. A lot of your glycoproteins are going to be things involved in composition and things between different cells. You can also have some glycolipids, so not everything is going to be proteins. But the vast majority of your cell membrane is going to be made up of phospholipid and proteins. Make sense? And those proteins are going to have a whole bunch of different kinds of functions. So some of the functions of membrane proteins are things like transport, which we're going to talk a whole bunch more about. We're going to talk about transport proteins. We're going to talk about carrier proteins and channel proteins. Those are both kinds of transport proteins. Does that make sense? So when we talk about those are kinds of transport proteins, which are going to be involved in allowing things to pass across your plasma membrane. Because remember, your plasma membrane is going to be semi-permeable. Semi-permeable means that some things are going to get through and some things are going to get stopped. Make sense? Okay. So part of that semi-permeability comes from these transport proteins, which kind of help regulate what's coming in and what's going out and what's not coming in or going out at all. You also have enzymes. So you can have enzymes that are associated with your plasma membrane. We talked a little bit about enzymes because enzymes are proteins. Enzymes are going to catalyze specific reaction. They're going to reduce the activation energy. You need to have some particular reaction take place. It's going to manipulate the substrates, the reactants, into a way that is more favorable for them to be converted into the products. Your prokaryotic cells, remember we said that they don't have the internal membrane-bound organelles. One of the ways in which they still carry out a lot of their functions, things that are in organelles in your eukaryotic cells, are by these enzymes located within their plasma membrane. So your prokaryotic cells still do cellular respiration. They're still making ATP. Some of them do photosynthesis. So they need to have the enzymes necessary for these reactions. They don't have the organelles. What they have are regions where they're kind of clustered in their plasma membrane, where these are the enzymes for this kind of pathway. Does that make sense? Okay. Signal transduction. today. We're gonna talk about the receptor is gonna interact with its signaling molecule. We're gonna get a interaction, potentially a conformational change, and it's gonna trigger a whole exciting pathway of events. That receptor is often a protein. We also have cell-cell recognition, where I said those glycoproteins, those proteins that are associated with carbohydrates, are often involved in cell-cell recognition. There they are. So sometimes your cells need to know what kinds of other cells are around them. It has your cell of a particular kind is going to have particular kinds of surface molecules, like your glycoproteins, that indicate what kind of cell they are. And so that helps your cells interact with each other. You have cells proteins that are involved in your intracellular journey, those junctions. What kind of junctions kind of have proteins that were filling the space in between them? Remember? The tight junctions, we had the proteins that were in between, right? So you can have proteins that are going to be associated with membranes that are going to be in between. And then you also have proteins that are involved in attachments to the cytoskeleton and the extracellular matrix. Proteins that are involved in either cells being able to move or be anchored in a particular location. Those are going to be often attached to the extracellular matrix. You'll also have proteins that are involved in attachment between the membrane and the skeleton to help for structure and movement within the cell. Okay. So let's talk a little bit more about some other features of the plasma membrane. So we said that it was a phospholipid bilayer, and that it had associated proteins, right? And that it was semi-permeable. Another way we describe it is that it's a fluid mosaic. So you have your phospholipid bilayer, you have your proteins, both the intrapole that's going to be associated with both leaflets and the peripheral that's associated with one or the other. And they don't just stay in one spot. Like if you're thinking about your cell, and we're thinking about, say, one of those exciting glycoproteins that's a way in which other cells know what kind of cell I am. That glycoprotein doesn't just sit at one spot in the plasma membrane, just stationary areas in one spot all the time. You have movement within your plasma membrane. Things can, like the protein, can move across the surface of your cell. They can move within that plasma membrane. The plasma membrane isn't like a super solid structure. We can describe it as a fluid mosaic. The phospholipids and whatnot that are making up your phospholipid bilayer have some ability to shift and move, which is how things like the proteins can move around the surface of your cell. It's also how you can get fusion between your different membrane components. So we talked about fusion before when we were talking about your cell and organelles, right? We said that the transport vesicle was going to butt off, and then it was going to fuse with the cisternae of the Golgi, right? Or we talked about during phagocytosis, that the food vacuole was going to get formed when we pinched in what we were trying to bring into our cell, and then it was going to fuse with the lysosome, right? Membranes, this phospholipid bilayer, they have this mobility to them, and it also allows them to fuse with other membranes and just become more, right? So there's a cool experiment where you have a mast cell, a mouse cell with their exciting membrane proteins, and you fuse it with a human cell, exciting membrane proteins. And what you get at the end is a now larger membrane sphere, but it contains both the mouse proteins and the human proteins, and after a relatively short amount of time, they're very much mixed throughout. It's not this side is the mouse proteins and this side is the human proteins. After a short amount of time, because you have that fluidity of the membrane, they're all mixed together. Does that make sense? This idea of a fluid membrane? The amount of fluidity of your membrane is going to depend on a variety of factors, because sometimes you need lots of mobility in your plasma membrane. We need to be able to move those enzymes or those surface molecules all around our cell easily. And sometimes we need our membranes to be a little more structured, and we need things to kind of stay where I put you, because perhaps you're going to interact with something else and we can't have you floating off, right? So depending on the kind of cell, depending on the function of the membrane, we're going to have variability in the fluidity of the membrane, in how much movement we can have in our membrane. And what is going to help regulate that fluidity, how much things are able to move around in our membrane. Remember back when we were talking about the plasma membrane, remember back when we were talking about lipids, and that, first of all, that they weren't polymers, and that they did have those exciting fatty acids, right? And in our phospholipids, we had a hydrophilic head, and then we had the phospholipid. Then we had the fatty acid tail, and those fatty acid tails could either have saturated or unsaturated fatty acids, right? And in our unsaturated fatty acids were the ones that had the double bonds, right? They were not fully saturated with hydrogens. They needed less hydrogens because they had the double bonds, right? So if you had the unsaturated fatty acids, then they had that little kink in them. And if you have the kink in the fatty acid tail, it's not going to be able to just butt up close against its neighbor, right? Because its tail is going to need more space. It's got the kink in it because of that double bond. So, if your membrane has more unsaturated fatty acids in it, you are going to have a more fluid membrane, because your phospholipids making up your bilayer are not going to be packed together, right? Everybody, the unsaturated guys need more space, as opposed to a membrane that has many more saturated fatty acids. They get packed together more tightly. It's a more viscous membrane is how we can describe it. It means you're going to have less movement in the plasma membrane. And so depending on your kind of cell and what's going on and what you need, the function, you have different amounts of the saturated versus unsaturated fatty acids. The other thing that can impact your membrane fluidity, if we're talking about animal cells, is cholesterol. So cholesterol is related to animal cells. Just so things like, fun fact, mushrooms or fungus, there's no cholesterol in mushrooms. But cholesterol, what its true function is to help regulate the kind of fluidity of membranes. So cholesterol allows you to regulate your fluidity kind of based on other conditions, not just the content of your saturated versus your unsaturated fatty acids. Cholesterol kind of acts as a buffer, letting you change your fluidity based on temperature. So in high temperatures, you have reduced fluidity, and you have the structure of the membrane. So again, key points is that phospholipid bilayer. It's got those hydrophilic head groups that are going to be pointing out into the environment and in towards the cytoplasm of the cell. In between are going to be those fatty acid tails. Those fatty acid tails are going to be either saturated or unsaturated, and that's going to influence the fluidity as well as cholesterol and help influence the fluidity in our animal cells as well. We're also going to have proteins. We're going to have integral proteins that are going to go through both leaflets. We're going to have peripheral proteins that are going to be associated with one or the other. And these proteins carry out a bunch of different functions: transports, enzymes, recognition, cell junctions, all kinds of fun things that they can do. Okay? So now, the next big kind of concept is going to be transport. It's going to be moving things across the membrane. Remember, our membrane is microbial. It acts as a barrier between the outside of the cell and the inside of the cell. It's going to help regulate what's going out and coming in. So, we're going to start with diffusion and osmosis. So your water and small non-polar molecules can diffuse across membranes. That was in your book, right? People remember water and your small non-polar molecules can cross membranes. So they don't need a transport protein. They're going to be able to move down their concentration gradient without, you don't need any additional energy put into the system to make that happen. Simple diffusion. Okay? So the molecules that can do simple diffusion are going to be your water and your small nonpolar. Nonpolar are going to be your hydrophobic molecules. Does that make sense? Okay. So your small nonpolar in the water, they can transport across your membrane, and that is the rate at which they do so. So they can. And kind of to give you context of the rate at which this is. So oxygen and carbon dioxide, that is relatively quickly, right? Because if you think about like a centimeter per second, that's like pretty quick, right? And consider that the size of your cell is like considerably smaller than a centimeter, right? If you can see a centimeter, you can't see a cell. So these molecules can go relatively quickly. It's water. We'll talk about when water can cross over the membrane via simple diffusion. Really, you don't see all of your water crossing over by simple diffusion. It can. That's not too slow. But we'll talk about a transport protein called aquaporins. Aquaporins are going to be how more of your water is going to transport across the membrane because it's going to be much faster than this. But, I mean, even water is still faster than, say, sodium and potassium, which are charged particles, which are not small, they're small, ions are small, but they are not nonpolar, and they are charged. And you can see that they take much longer to cross over the membrane. Does that make sense? So all of these are kind of talking in a relative way. Does that make sense about simple diffusion, about what can get across and what can't? Okay. Cool. So, as it said, diffusion is going to be this using the energy of the concentration gradient to move. Okay? So there is, if you remember, potential and kinetic energy, right? You have potential energy is the energy of position, kinetic energy is the energy of movement, right? gonna power the transport for things to move down their concentration gradient. Does that make sense? Okay, so then, my exciting picture here, I have the orange bulbs, I have a separation, and now I have more on one side than the other, right? Okay, here we go. Okay, so here I have set up a concentration gradient. And so what's gonna happen? They're gonna move down their concentration gradient. So here we are having our simple diffusion because it's small, non-polar molecules. They are going to move down their concentration gradient using the energy of the concentration gradient. The potential energy of the concentration gradient is powering this movement. Make sense? It's also why it's not going to go on forever. So I'm not going to move all of the orange balls from the left side to the right side. Because, so they're going to move from the area of higher concentration to lower concentration. But ultimately, I'm going to get to a point where I have an equal number on both sides, right? So I have them all on the left, they start moving to the right. At some point, I'm going to have an equal number on the right and the left, right, as they're going down the concentration gradient. Once that happens, once I now have an equal amount on the right and the left, I don't have a concentration gradient anymore. I don't have that potential energy that comes from having more on one side versus the other. So I'm not going to get any more net movement. They're not going to all just stand still. But for every one that goes right, I got one that's going left. Does that make sense? So you reach what's called an equilibrium. So your diffusion is going to be powered by the potential energy of the concentration gradient. It requires that concentration gradient. The way that these small nonpolar molecules, they want to be able to go with no energy input. If they want to be able to just move, they require that concentration gradient. Does that make sense? And each solute, each thing that we are talking about is going to have their own concentration gradient. So it's not like total number of solutes or total number of particles outside and inside of your cell. Everything's going to have their own concentration gradient. If I have purple balls and orange balls, the purple balls have no interest in the orange balls have no interest in the purple balls. They're each going to go down their own concentration gradient until each of them reaches their own equilibrium. Okay. So, we're feeling good about diffusion. Okay? So that was simple diffusion of my super cool small nonpolar molecules. So now, we're going to talk about osmosis. And what osmosis is the diffusion of water. If you've had lab, you've had some discussion of osmosis already. If you have not had lab, you will have a whole discussion about osmosis when you do get there. What osmosis is the diffusion of water. It's water moving down its concentration gradient until we get to that equilibrium. When water is moving down its concentration gradient, another way you can think of it is going to be moving towards the area of higher solute concentration. Because if I have more solutes, then I have less water. If I have less solutes, then I have more water. Does that make sense? That's kind of key to understanding osmosis as the diffusion of water. If I have more solutes, I have less water. If I have fewer solutes, I have more water. Does that make sense? Okay, so then you can remember it the same way that you're remembering diffusion. It's going to move down its concentration gradient. So here I have okay. So let's say I have membrane is gonna let water through, but it's not going to let the balls through. A little solute, a little red balls. Okay? So what's gonna happen? In this situation, water is gonna move. So I have a different amount of solute on the two sides of my permeable membrane, right? Water, because it's gonna be, the membrane is permeable to water. Water can pass through my membrane. The water is gonna move. Which way is the water gonna move? In my drawing here. The water is going to move this way. The water is going to move towards the area of higher solute concentration. It's going to move towards the lower water concentration. Does that make sense? And so what am I going to see when that happens? I would see the water level go up, right? Because water is going to come from here, and this water level is go down, right? Does that make sense? My water is going to move, in this case, from the right side to the left side. Because water can get through. The little membrane that I had was not permeable to the little red balls. So the little red balls are going to be stuck. They can't switch sides. If the little red balls could move, like if this membrane was permeable to both water and red balls, the red balls would also diffuse from left to right, so that they would diffuse down their concentration gradient, right? But in this case, I have that membrane, and only the water can move, right? So here, the water is moving, the water is gonna move down its concentration gradient, it's gonna move towards the higher concentration of solutes. Does that make sense? Maybe the opposite. I had a 50-50 shot. All right, so osmosis, diffusion of water. Okay. Okay. So that will also relate to the concept of tonicity. So tonicity is the ability of a surrounding solution to cause a cell to gain water. Tonicity is going to be a description of a solution in relation to a cell. Okay? It's important to understand that. It's the relationship between the solution and the cell that's within it. And it's going to be a description of the solution. If you change the cell, you can change the relationship. That same solution, if we have a different cell, then it's going to potentially have a different relationship with that cell, and it would change the tonicity of that solution. Does that make sense? Because it's this relationship that kind of is described by tonicity. So the tonicity of the solution is going to depend on the concentration of the solutes that can't cross the membrane relative to its cell. So, do this study again. Okay. All right. So, if I have a cool cell in a solution, and my cool cell has 3 solutes in it, and I have in my exciting membrane, and it's probably going to burst when all this new water shows up. If I have a plant cell though, this is how plant cells like to be. Because if you remember, plants do not have bones. Plants like to be very full of water. They like to have their cells very full of water, and their plasma membrane just pressing up against that cell wall, because it helps kind of give them some structure. So that's how plant cells like to be in hypotonic solution. If I had my same cell, it's exciting green cell use, and I had it in a solution with 4 solutes. So now I have a higher concentration of solutes in my solution than I had in my cell. Where's the water gonna go? It's gonna go out of my cell, right? This is hypertonic. Because the water is going to go down its concentration gradient toward the area of higher solute concentration. My cell is going to shrivel. If I'm talking about my animal cell, my poor animal cell is just going to shrivel up. If I'm talking about my plant cell, that plasma membrane is going to start pulling away from the cell wall. And then an isotonic is when I have an equal amount on both sides. And this is how my animal cells like to be. My animal cells like to be equal on both sides. For all the water coming in, we got water going out. We're at like equilibrium there, right? So for all the water that comes in, an equal amount goes out, and we just stay zero. We have an equal amount of solutes on both sides. Make sense? There we go. And there's a picture. As I said, what the result of this water moving in and out, the impact of this tonicity is going to depend on the kind of cell we are looking at. Your animal cells want to be in an isotonic solution, whereas your plant cells want to be in a hypotonic. They want water moving in, whereas animal cells want water moving in and out equally. Okay. So that brings us to facilitated diffusion. So both simple diffusion and osmosis can happen without any transport proteins. Okay? You can have your small nonpolars diffusing across membranes. You can have your water moving across your membrane for osmosis. Slowly, but it can do it. Depending on its concentration gradient with no additional energy needed. Facilitated diffusion is going to be your passive transport aided by proteins. So in this case, your facilitated diffusion is still going to be powered essentially by that potential energy of the concentration gradient. But we are not moving one of our small nonpolar molecules. We're moving larger molecules. We're moving ions, charged particles. They can still use the power of the concentration gradient to move across the membrane, but they're going to need to do it through a transport protein. They are going to need to go through the transport protein, which helps regulate what's getting in and out of our cell. These transport proteins are really important towards the idea of that semi-permeableness. So in your facilitated diffusion, we need a transport protein. They can be either a channel protein or a carrier protein. What's the difference? They're both transport proteins. Your channel proteins are going to be essentially like they're gonna go just through the plasma membrane. They are specific. All of your transport proteins are gonna be specific. They're gonna regulate what's getting through via shape, because they're proteins, and remember, shape is really important to their function. Whatever they're supposed to let through is gonna have the right shape that it can go through. That's how you get that selective permeability. So a channel protein is gonna be a channel that things are gonna be able to go based on the concentration gradient in and out of the cell. Your carrier proteins are going to have a shape change. So they're going to interact with whatever their solute is, and it's going to cause a shape change, and that's going to let it cross over the plasma membrane. So in my little example here, you have your solutes. They want to move down their concentration gradient. I have more outside the cell than I have inside of my cell. Maybe more in the blue than in the brown. They're going to be able to move down their concentration gradient. They're going to interact with my carrier protein here, and then when they interact, I get a shape change. It's going to let them into the cell. Does that make sense? So in facilitated diffusion, when we're moving down the concentration gradient via these transport proteins, they can be either your channel proteins or your carrier proteins. That's important. So an example is an aquaporin. Aquaporins are the transport molecule for water. They're an example of a channel protein, and they are specific for water. They're like the channel protein for water, and they will let your water move in and out of the cell down its concentration gradient. Make sense? You have a whole bunch of these ubiquitously throughout all kinds of cells and tissues throughout your body, because your cells need water. Active transport is going to use transport proteins. It's only going to use carrier proteins. So here, this is why this was important, right? Facilitated diffusion can use either carrier or channel proteins. Active transport is going to need a transport protein. It always is a carrier protein. All of your transport proteins for active transport are going to be carrier proteins. They're going to have that shape change involved. Your active transport is going to move things against their concentration gradient. So here, I need to move things from areas of lower concentration to areas of higher concentration. I'm going against the concentration gradient. If I'm going against the concentration gradient, I cannot use the exciting potential energy of position of the concentration gradient. If I'm moving against the concentration gradient, I'm going to have to put energy in from an outside source. So that's where ATP is going to come in. ATP is going to provide that energy to transport things across their concentration gradient. It's going to power that shape change in my transport protein so that I can move against the concentration gradient, so that I can move things in or out of the cell against the concentration gradient. That shape change that it depends on is going to get powered by this ATP that I'm putting in. It's the reaction. Does that make sense? So here I have an exciting example. It is the sodium-potassium pump, which you've probably heard of from cells, right? The sodium-potassium pump, that you have a higher concentration of sodium outside of your cell than inside. You have a higher concentration of potassium inside than outside. But we need to transport your sodium out and your potassium in. So that's against the concentration gradient. And so in order to do this kind of transport, these carrier molecules have to have ATP put in. And what it looks like is you have a binding of your solutes, your sodium. You have ATP. It hydrolyzes. We'll talk about more about that at a future time. theoretical, but again, the key take home being that the sodium potassium pump is an example of active transport where things are gonna move against their concentration gradient. And if you're moving against that concentration gradient,